everyone, and welcome to the second class of this week. We will talk about taphonomic factors and the effect that they have in bones. Before we start talking about the taphonomic factors, it's important to know what is taphonomy. And when talking about taphonomy in the field of anthropology and archaeology, it refers to the study of what happens to the body after death. It involves the decomposition of the body that is especially important to forensic anthropology to estimate how long the individual died. And then there are the study of the taphonomic factors applied only to the bones, and we are going to focus only on this part. The taphonomic factors can be divided into intrinsic and extrinsic. The intrinsic are related to the individual and the extrinsic to the environment where the individual is buried. Within the intrinsic taphonomic factors is the size of the bones, the shape, the density, the age of the individual, pathologies, and the sex. In the case of the extrinsic factors, they can be divided into environmental, including water, soil type, temperature, and air, then flora and fauna, and the man action. And we are going to talk about each of these factors individual, but they never act alone. Starting with the size and the shape of the bones. So in individual burials, the larger bones are the ones that tend to be more preserved because there aren't other bones of other individuals that can be damaging these ones. On the other hand, in collective burials, the smaller bones are the ones that usually are better preserved because they are less susceptible to be affected by other individuals. So in this case, you have to think about the time when the individuals were buried. For example, in a collective burial, if two individuals are buried on top of the other, the bones of the individual that is under the other are taking a lot of weight that can, for example, break the bones. And in this case, the bigger ones are more susceptible because they have a larger available surface. Looking at the shape of the bones, the ones that have an irregular form are usually less preserved because they don't resist very well to the weight of the soil. In this image here, you can see that both the cocci are fragmented, and their form is more irregular than a tubular one. On the other hand, the bones of the arms are well preserved once their shape is tubular. As you can see here, this humerus is very well preserved. Another factor is the bone density. As we talked in the first week, there are two types of bone. The spongy that has a lot of pores and the compact bone that only have microscopic pores. This is important because the bones that have higher proportions of spongy bone are more susceptible to taphonomic factors than the ones that have more compact bone. As the spongy bones have a lot of pores, it is more fragile. For example, in the long bones, the diaphysis is usually better preserved than the epiphysis, because the diaphysis is mainly compact bone and the epiphysis spongy bone. As you can see in this image here, you see this tibia and you see that the diaphysis is very well preserved. But if you look at this part of the bone, that is the epiphysis, you see that it is damaged. Age at death is also an intrinsic factor because the bones of the non-adults are more fragile. And this happens because their mineral composition is lower than the mineral composition of the bones of the adults. In the case of sex, the women suffer more of osteoporosis, which is a disease that reduces the mineral content of the bones, making them fragile. Also, in general, the body size of a male individual is bigger, leading to bigger bones. And finally, the funerary practices. In the past, the graves of the males were more elaborated than the females, giving more protection to the skeleton. The last intrinsic factor are the pathologies. They can have a positive or a negative impact in the preservation of the skeleton. If a pathology responds by forming bone, it makes the bones more resistant. On the other hand, if a pathology destroys bone, it makes the bone less resistant. We will talk more about the responses of the bone to pathologies, but to give you an example, In this image here, you see a pathology that is called dish that forms bone, as you can see here in this part of the vertebrae. And eventually, the vertebral column fuses. And in this case, these bones are more protected to taphonomic factors. On the other hand, in this image here, you can see also vertebrae, and this vertebrae has brucellosis that makes these holes that you can see. So these holes makes the bones less resistant. So moving on to the extrinsic factors, the firsts are the water and the soil type. The water is one of the most influential agents of the extrinsic factors. It leads to leaching of the bones, making them fragile. When thinking about water, it's important to note the humidity, the precipitation, and drainage. And the drainage is very important because if bones stay in a place full of water, they are going to be in a very bad condition. And then the soil type. Usually, acidic soils lead to a worse preservation, and neutral or basic to a better preservation. The amount of salt in the soil has also an impact in the preservation of the bones. Soils with high contents of salt poorly preserves the bones. Also, if a burial is superficial, the bones are more susceptible to soil erosion. And finally, in the soil type, the pressure of the soil can lead to a faster decomposition. The temperature varies with latitude, season, and depth of the burials. The bones are better preserved in places where the temperatures are mild and don't vary much. The depth of the burial is important in this topic because deep burials tend to preserve bones better because this way they are less subject to wide temperature ranges. And then the oxygen. The oxygen have a higher impact in the decomposition of the body than in the bones, and more oxygen leads to a faster decomposition. So in these images that you can see, they are actually animal bones, but the animal bones also suffer from taphonomy as well as human bones. So you can see that both of these fragments of bones have cracks, as you can see, for example, here, and then this one here. And these cracks are taphonomic changes that were caused by both the contact with water and wide temperature ranges. So now we are going to see the effects of the flora and the fauna. And starting with the roots, they often leave multidirectional grooves in bones. As you can see in this image, these grooves here were all caused by roots. And also in long bones, the roots can get inside the medullary channel and making the bone more prone to break. The animals frequently modify human remains. Rodents and canines can leave the edges of the bone gnawed. You can see here in this image, this is the skull of a deer, and this part here that you can see in the tail has marks of the rodents. Scavenger birds can also leave signs of their beaks. These holes here in this bone and this hole here were caused by an eagle. And finally, the insects. The insects can destroy bone by creating pulpal chambers, as you can see in all of these fragments of bone. These are human bones, these two are the distal end of the humerus, and these holes that every fragment of bone has were caused by insects. And finally, in the extrinsic factors, the man action. So we are the ones that decide who is buried, where that happens, and how that happens. All these decisions have an impact in the preservation of the skeleton. For example, if we decide to inhume an individual in a place that has a lot of water and another in a place that has no water, the bones of this one will be more preserved. And as you can see, all the taphonomic factors are interconnected and we can think about them separately. So lastly, it's important to think of what it is the impact that these taphonomic factors have in the study of human remains. The more complete the skeleton is, the more information it's possible to extract from them. When the skeletons are very fragile and fragmented or damaged, There will be a lot of specific parts of the bones that are important to apply certain methods to estimate the biological profile that aren't present or are damaged. So with less information about the individuals, it's harder to study a population. In the next week, we are going to talk about diets, pathologies, and how the study of human skeletons contributes to cultural heritage. If you have any questions, please let me know, and I will see you next week.